Click the bell icon and subscribe to our channel so that you will not miss any classes. Hi everyone, let us start with a new lesson, that is Biomolecules. This chapter is important for both NEET exam as well for board exam. Now let us quickly start the lesson and learn as much as possible. When an elemental analysis is done for a microbial paste, animal tissue or a plant tissue, we will obtain the same list of element, and the list goes like this. It contains hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, etc. Same if we done for the earth crust, we will obtain the same list of element, but the difference is there is a relative abundancy of hydrogen, carbon, oxygen and nitrogen in human body as compared to the earth crust. When you look at this table, we will observe hydrogen composition is 0.5% in human body and that of earth crust is 0.14%. Carbon is 18.5% and that of earth crust is 0.03%. Oxygen percentage is 65.0% in human body and that is 46.6% in case of earth crust. Nitrogen is 3.3% in human body and that is very little in earth crust. So we can draw a line here. In a textbook also you could do the same. If you draw a line here, there is a relative abundance of hydrogen, carbon, oxygen and nitrogen in human body and that is less in case of earth crust. When you look at sodium and sulfur, the concentration is less in case of human body and that is more in case of earth crust. Remember, this table is very important for competitive exam. They may ask about the element and their percentage respectively. Now, how to analyze a chemicals present in a living tissue, and the analysis could be done for the chemical compounds present in them, especially the organic compounds. For this, a living tissue have to be taken and that tissue could be vegetable piece or a liver tissue. So you could ask whether you have to take only this or any other. You could take any other. In NCERT they have mentioned it, that's why I took this. Living tissue has to be ground by adding a chemical trichloroacetic acid. We will obtain a thick slurry and this thick slurry should be filtered using a cheesecloth or a cotton cloth. Now we are adding this slurry to the filter and here we have placed a cheesecloth or cotton cloth. When we filter, the two fractions we will obtain. One is called filtrate and here whatever present is called retentate. Now in filtrate and retentate, there is presence of many chemical compounds. Let us analyze what are the compounds present in filtrate and retentate respectively. In filtrate, the filtrate is more technically called as acid soluble fraction. This is very important. This acid soluble fraction contains thousands of organic compounds and these thousands of organic compounds have the molecular weight ranging from 18 to 800 Dalton and they include simple sugars, nucleotides and amino acids. Along with this, there is presence of many inorganic compounds also, for example sulfates and phosphates. One more thing you have to study here, these organic compounds which are ranging from 18 to 800 Dalton are called microbiomolecules. This is purely based on their molecular weight, nothing else. Now, in retentate, whatever present here, the retentate contain a acid insoluble substances. Hence, it is called acid insoluble pool. More technically, it contains 4 types of organic compounds in it. They include proteins, nucleic acids, polysaccharides, and lipids. Among these 4, proteins, nucleic acids, and polysaccharides, they have the molecular weight more than 1000 Daltons. Hence, they are considered as macrobiomolecules. Here, lipids, even though they are separated in acid insoluble pool or in retentate, they are not strictly a macrobiomolecules because lipids are not present freely in a cell. They are present in a structures, especially cell membranes. Now, when we grind the tissue, these lipids will form a vesicles. And vesicles are water insoluble, hence they are separated in the acid insoluble fraction. Hence they are not strictly called a macrobiomolecules. And one more point you have to remember here, when an element compound analysis is done for a living tissue, we will have the filtrate and the filtrate will roughly give a compounds which are present in a cytoplasm. This is very important, they may ask it. And in retentate, it contains both cytoplasmic content as well as it contains the structural components of organelles. This point is very important. Now, all these whatever I explained here, it is from introductory part, and about microbiomolecules, macrobiomolecules and their molecular weight, and lipids, why they are not strictly a macrobiomolecules. It is given under the heading macrobiomolecules in NCERT textbook. So you can, whenever you are reading, you can refer first introductory part, and immediately after analysis of compound you could go for macrobiomolecules. There you will get all these. When we analyze a chemical compounds present in a living tissue, we will obtain a components which are present in the living tissue. The components present are water, proteins, carbohydrates, lipids, nucleic acids and ions in a varying percentage. Water is 70 to 90% of water, proteins 10 to 15%, carbohydrates 3%, lipids 2%, nucleic acids 5 to 7% and ions only 1%, which suggests that we need to have more and more water and more proteins in our food. So this table will be helpful for maintaining our diet chart also. Now, all these compounds, so we have got one list. For example, simple sugars, nucleotides, amino acids, proteins, nucleic acids, polysaccharides, lipids. And all these compounds are having the presence of carbon in them. Hence, all these carbon compounds which are obtained after analysis of a chemical compound for in a living tissue, then these carbon containing compounds are together referred as biomolecules. So we have roughly classified them as microbiomolecules and macrobiomolecules based on their molecular weight. 18 to 800 Dalton biomolecules are called microbiomolecules, and those are having the molecular weight more than 1000 Dalton are called macrobiomolecules. Now, we will have a group of compounds here. Now, how to analyze for a particular organic compound? A particular organic compound can be extracted by using various separation techniques, say for example chromatographic technique or centrifugation technique, any other things. Then it is subjected to analytical technique. After subjecting it to analytical technique only, we will obtain a molecular formula of that compound and the possible structure of that compound. So they may ask question like this: how molecular formula and possible structure of a compound present in a living tissue can be determined? It is determined by only after extraction and subjecting them into an analytical technique. So this is about the organic compounds which are present in a living tissue. Here, one more thing we will get interest, that how inorganic elements and compounds can be analyzed in a living tissue, whether they are present or not. Of course, organic compounds are present, along with that various types of inorganic compounds and elements are also present, that we came to know here itself. In filtrate there is presence of sulfates and phosphate. So likewise, there are other elements also present in the living tissue that can be analyzed by using a destructive method. Destructive method is called a ash analysis, where the living tissue, a small amount will be taken and it should be weighed. After weighing, it has to be completely dried. Drying is important because a living tissue contains more amount of water. So water has to be evaporated. For that, drying is essential. After drying, that tissue will get a dry weight, and that dry weight has to be fully burned. After burning, the carbon containing compounds are oxidized and they will be evaporated in the form of carbon dioxide. Whatever remaining is called the ash, and ash contain the elements like calcium and magnesium. So this will gives us the entire chemical composition of a living tissue. It contains both organic compounds and inorganic compound. The organic compound list goes like this: simple sugars, nucleotides, amino acids, proteins, nucleic acids, polysaccharides, lipids. And inorganic compounds list goes like this: sulfates, phosphate, calcium, magnesium, and etc. So this is all about the introductory part of biomolecules. In next class, let us discuss in detail about amino acids, simple sugars, lipids, and what more all. And thank you. Be waiting for the next video.